Neurobiologist Robin Cooper and grad student Cole Malloy use fruit flies and crayfish to illuminate the complexities of the human brain.
The power of genetics allows us to work with the Drosophila and do things really you can't do with any other organism, in the sense of rapid development, manipulate genes very quickly, and test out many different aspects from behavior to how the neural circuits are formed. And you can relate whole animal behavior to the mechanisms behind that behavior. In particular, we're interested in synaptic transmission, and our past NSF funding has been looking at the differences in high output and low output communication between synapses, such as when a nerve communicates to a muscle or to another nerve, there are differences in the synaptic structure and the proteins involved. Some of the most exciting results we've had recently was with some new techniques that we started in the lab, and it's just gone every direction actually. It's called optogenetics, and it's where you can take a gene from, say, a blue-green algae, and people have been able to put that into the Drosophila genome, and that gene expresses a protein that is sensitive to light. These give us a lot of power of, in the intact animal, you can shine blue light on the animal and then activate just serotonergic neurons. So instead of using pharmacological approaches, we can use more of an intrinsic approach, activate that neuron, and then see how that changes the animal's behavior.
We study how the nervous system adapts to changes in the environment, and what kind of influences that change are various neuromodulators. And I actually study a neuromodulator called acetylcholine that's prominent in humans as well as in fruit flies. My job for the past couple of years has been kind of characterizing those receptor subtypes using multiple techniques, from an optogenetic approach to pharmacological approach, and then also to kind of identify how altering acetylcholine signaling in the brain throughout development then alters formation of neural circuits. One exciting thing that we found recently is that GAD1 is an enzyme called glutamic acid decarboxylase. It actually converts a neuromodulator glutamate to GABA, and we found that there might be expression of this enzyme in skeletal muscle and cardiac tissue in flies. And that's kind of interesting because what's been published in the past is that this enzyme is probably just expressed in the nervous system. So that's important because there may be some off-target effects using GAD1 to drive expression of various proteins in the nervous system. You might be getting actual expression in muscles too. So that's kind of interesting. I think the breadth of the field is what kind of drew me to biology. In high school, I was more a chemistry guy, like my advisor. And when I came to the UK, I became more interested in biology, especially with genetics and neuroscience. And I think the genetics and neuroscience aspect comes from my background as being an identical twin. But I've kind of always been interested in what makes my brother and I individuals. What about our nervous system development and how our environment has shaped our nervous system and how we have different and unique personalities given the fact that we are genetically identical. So yeah, that's kind of always drawn me to understanding how the brain works.
Now we're looking at it at a basic science level, but you think about maybe a mother that just gave birth and she's undergoing postpartum depression, and she needs to take an antidepressant like fluoxetine or Prozac. We know that Prozac will get into the breast milk. But what are the impacts on that fetal development? And these are questions that people haven't really addressed yet fully. If a serotonergic neuron is stimulated and it's releasing a lot of serotonin, if the target cell doesn't branch as much because it's getting so much activity, that's a structural change that we can pick up. With optogenetics, we're able to really target these particular neurons like serotonin, dopamine, acetylcholine, look at changes in behavior of the animal, as well as developmental aspects.